Hello. So today we'll be talking about one of the questions that came in GATE 2023 Biotechnology paper. DNA sample collected from an unidentified bacterial species Y contains 13% of adenine. The G plus C or the guanine plus cytosine content in percentage of Y is. So before solving the question, we'll learn a little bit about Chargaff's rule, which basically led to the DNA double helix structure that we know of today, and we'll see how this rule can help us solve this question. So the discovery of structure of DNA involved several people and highly interdisciplinary involvements. Rosalind Franklin and Wilkins along with Gosling acquired X-ray diffraction images, James Watson and Crick developed the models based on the available information, and finally deciphered the complete structure. Yet a very important conclusion about the structure of DNA was made on the foundations led by Edwin Chargaff. Today, what we know as the Watson-Crick pairing in DNA, that is represented as AT or GC pairing, was only possible after the enormous efforts of this scientist named Erwin Chargaff, who spent several years of his life contributing to the basic science and understanding the base pair composition of DNA in various species. Okay, so what are the Chargaff's laws, and how do these Chargaff's leading discoveries that we today know as Chargaff's rules, how these rules basically helped in the forms the foundation of Watson-Crick base pairing in DNA. Okay, so the first parity rule states that in any double-stranded DNA, the number of guanine units equals the number of cytosine units, and the number of adenine units equals the number of thymine units. So the rule establishes the pair of A, T and GC, and this enabled the Watson and Crick to finally complete the DNA double helix model. Okay, so the percentage of A is equal to the percentage of T, and the percentage of G is equal to the percentage of C. Okay, so there are several problems that come in exams, such as determine the percentage of adenine if the total cytosine content is 20%. This can be solved by using this first parity rule, which implies that if cytosine is 20%, then guanine will also be 20%. Then the remaining 60% will be A and T pair, hence each one of them would be 30% each. So this also implies that the A plus G and the T plus C ratio for an organism shall be nearly equal to 1 or exactly 1 in most cases. Okay, so this is what Chargaff's law is all about, and this law will help us solve this question. So if you can see this slide also, we all know about the DNA double helix structure. This is the DNA double helix structure, and we all know that DNA has the double helix has two DNA strands, antiparallel two DNA strands, and there are sugar phosphate backbones, and the base pairs form hydrogen bonds with each other. So C and G, that is cytosine and guanine, form 3 hydrogen bonds, while adenine and thymine form 2. And this is how the antiparallel DNA strands come together to form a DNA double helix. So the pairing of nitrogenous bases is a very important factor in forming the DNA double helix structure, and this also sits well with Chargaff's rule, which say that the percentage of A is equivalent to the percentage of T, and the percentage of G is equivalent to the percentage of C. So if we move to the question that we have here in the GATE 2023 Biotechnology paper, so the percentage of adenine is 13% that they have mentioned in the question. So that means that the percentage of thymine will be 13% approx. Okay, and therefore the percentage of adenine and thymine will be 13 plus 13 or 26%. So the A plus T will be 26% total. Now the total of all the bases is 100%, that we know. That means adenine, thymine, guanine and cytosine, all of these together forms the 100%. So therefore the percentage of G plus C or guanine plus cytosine will be 100 minus A plus T, which is 100 minus 26, which is 74%. So this is how we come to the answer 74%. So the question was DNA sample collected from an unidentified bacterial species contains 13% of adenine. The G plus C content of Y will be 74%. So this is how we can use Chargaff's rule to solve these type of questions. If you want to learn more about Chargaff's rule, the DNA double helix model, then you can actually refer to this course, NPTEL course, Proteomics Principles and Techniques by Professor Sanjeeva Srivastava. So there is a module 2, lecture 2 that talks about the central dogma, basics of DNA, RNA and proteins, and in this lecture there is a good explanation of Chargaff's law and how basically that can be interpreted, and this law can be used to solve these kind of questions in the GATE papers. Thank you.